In 2019, a team of geneticists at the Broad Institute completed what many thought was impossible. They mapped the brain tissue DNA of over 15,000 individuals from every major population group on Earth: Europeans, Africans, East Asians, South Asians, Indigenous Americans, Pacific Islanders. The largest study of its kind ever conducted. The goal was simple: find out if the genes controlling brain development, cognitive function, and neural architecture differed meaningfully between racial groups. The results arrived 3 years later. When the lead researchers presented their findings at the annual genetics conference in Boston, the room went silent. Not because they found what people expected, but because what they discovered completely dismantled one of the most persistent assumptions in human history: the idea that race determines intelligence, that certain populations are genetically predisposed to be smarter, faster learners, better problem solvers. The data showed something else entirely, and the implications of what they found are still sending shockwaves through genetics, anthropology, and neuroscience today. If you want to see more deep dives into genetics and ancestry research that challenges what we think we know, hit that subscribe button right now. I cover breakthrough studies like this every week, breaking down the science that rewrites human history. To understand why these results were so shocking, you need to understand what scientists were actually looking for. The human brain contains roughly 86 billion neurons. The genes that control how these neurons form, connect, and communicate are some of the most complex in our entire genome. Researchers focused on 3 main categories: genes involved in brain size and structure, genes linked to synaptic plasticity, which is how the brain learns and adapts, and genes associated with neurotransmitter systems like dopamine and serotonin that regulate mood, motivation, and cognitive processing. The assumption going into the study was straightforward. If racial groups had been separated for tens of thousands of years, evolving in different environments with different survival pressures, then surely the genes controlling brain function would have adapted differently. Cold climates versus tropical climates, hunter-gatherer societies versus agricultural societies, isolated island populations versus continental populations with constant migration and mixing. This wasn't a fringe theory. It was baked into centuries of scientific thinking. In the 1800s, researchers measured skull sizes to prove Europeans had larger brains. In the early 1900s, IQ tests were designed with cultural biases that made immigrant populations score lower. Even into the 1990s, books like The Bell Curve argued that intelligence gaps between races had a genetic basis. The science was always flawed, always shaped by the biases of the researchers. But the idea persisted. So, when the Broad Institute launched this study, the scientific community braced for controversy. Would the data confirm what racists had been claiming for centuries? Or would it finally put the debate to rest? The researchers started by sequencing brain tissue samples from deceased donors who had agreed to contribute to genetic research. They specifically focused on tissue from the prefrontal cortex, the region responsible for decision-making, problem-solving, and abstract thinking. This is the part of the brain that makes humans uniquely human. If there were going to be differences, this is where they would show up. The technical challenge was immense. Brain tissue degrades rapidly after death, so the samples had to be processed within hours. The team used single-cell RNA sequencing, a technique that allowed them to examine gene expression in individual neurons, rather than just averaging across tissue samples. This granular approach meant they could see not just which genes were present, but which ones were actively being used to build proteins and maintain neural function. They also examined the hippocampus, the brain region critical for memory formation and spatial navigation, and the amygdala, which processes emotions and fear responses, and the cerebellum, which coordinates movement and motor learning. Each region was analyzed separately to see if population differences emerged in specific cognitive domains. Maybe one group would show variants affecting memory, while another showed variants affecting emotional processing. The researchers wanted to capture everything. Next, they cross-referenced the genetic data with the self-reported ancestry of each donor. They grouped individuals into continental ancestry categories: African, European, East Asian, South Asian, Native American, and Oceanian. These categories aren't perfect. Human migration and mixing have blurred the lines for millennia, but for the purposes of large-scale genetic analysis, they provide a useful framework. The sample size was carefully balanced, equal numbers of individuals from each ancestry group to avoid statistical bias. Donors ranged in age from 25 to 85 at time of death, allowing the team to look for age-related patterns in gene expression. They also controlled for sex, since some cognitive traits show small sex-based differences in genetic influence. Every variable that could confound the results was accounted for. Then came the hard part, comparing the genetic variants across groups, not just looking for differences, but looking for differences that actually mattered. A single nucleotide change in a gene might look different on paper, but have zero functional impact. The team needed to identify variants that altered gene expression, protein production, or neural development in meaningful ways. They focused on what geneticists call coding variants, changes to DNA that directly alter the structure of proteins. These are the variants most likely to have functional consequences. They also looked at regulatory variants, changes in the regions of DNA that control when and how strongly genes are turned on. A gene might be identical across populations, but if one group has a variant that makes it express twice as strongly, that could matter. They ran the analysis and ran it again and again, triple-checking the results because what they were seeing didn't make sense. The genetic differences were there. Of course they were. Every human population has unique genetic markers shaped by thousands of years of evolution and adaptation. Africans have genetic variants that protect against malaria. Europeans have variants that allow lactose digestion into adulthood. East Asians have variants that affect alcohol metabolism. These differences are real, measurable, and scientifically documented. But when it came to the genes controlling brain development and cognitive function, the story was completely different. The variation within each racial group was far greater than the variation between racial groups. An individual from Nigeria and an individual from Sweden might have more similar brain gene profiles than two individuals, both from Nigeria. The genetic lottery that determines how your brain develops, how your neurons connect, how efficiently your synapses fire, that lottery doesn't care about your ancestry. The researchers found over 200 genetic variants associated with cognitive traits like memory, processing speed, and verbal ability. These variants exist in every population. Some individuals have more of the variants linked to higher performance. Some have fewer. But there was no pattern, no cluster of variants that appeared more frequently in one racial group versus another. The distribution was random. One of the lead researchers, Dr. Alicia Martin, put it bluntly in her published paper. She wrote that the genetic architecture of the brain shows no evidence of population-specific adaptations related to cognition. Translation: evolution didn't make one group smarter than another. It didn't happen. This wasn't the first study to reach this conclusion, but it was the largest, the most comprehensive, and the most methodologically rigorous. It used cutting-edge sequencing technology that earlier studies didn't have access to. It controlled for variables that had plagued previous research, and it included populations that had been historically underrepresented in genetic studies, particularly indigenous groups and people from the global south. The findings aligned with decades of research in evolutionary biology and anthropology. Modern humans, Homo sapiens, emerged in Africa roughly 300,000 years ago. Small groups began migrating out of Africa around 70,000 years ago. In evolutionary terms, that's a blink. Nowhere near enough time for significant cognitive differences to evolve, especially when you consider that the pressures shaping brain evolution, things like social cooperation, language development, and tool use, were universal across all human populations. To put this in perspective, it takes roughly 1 million years for a new species to evolve through natural selection under strong environmental pressure. The time since humans left Africa is about 7% of that time span. The genetic changes we see between populations, the ones affecting skin color and disease resistance, those are relatively simple adaptations. Single genes or small clusters of genes under intense selective pressure. Brain development is the opposite of simple. It involves thousands of genes working in precise coordination. Mutations that significantly alter brain function are usually harmful, not beneficial. Evolution tends to conserve these critical systems rather than tinker with them. The genes controlling how neurons form, migrate, and connect during fetal development are nearly identical across all mammals, let alone across human populations. Consider the FOXP2 gene, sometimes called the language gene, because mutations in it cause severe speech and language disorders. This gene is so important that it's been conserved for millions of years. The version humans carry differs from the chimpanzee version by just 2 amino acid changes that occurred sometime in the last 6 million years of human evolution. But the version of FOXP2 in a person from Japan is identical to the version in a person from Kenya or Norway. 0 differences, because any change that disrupted this gene's function would be immediately weeded out by natural selection. The same pattern holds for hundreds of other genes involved in cognition. They're too important to mess with. Evolution has locked them in place across the entire human species. But here's where it gets even more interesting. While the study found no cognitive differences, it did find differences in other traits. Genes related to skin pigmentation, hair texture, and facial features showed clear population-specific patterns. This makes sense. These traits were under direct selective pressure from environmental factors like UV radiation and temperature. Darker skin evolved in equatorial regions to protect against sun damage. Lighter skin evolved in northern latitudes to maximize vitamin D production. The genetic changes are obvious and measurable. So why didn't the brain follow the same pattern? Why didn't cognitive abilities adapt to local environments the way skin color did? The answer lies in how evolution works. Natural selection acts on traits that directly impact survival and reproduction. If a genetic variant gives you an advantage in your environment, you're more likely to survive, reproduce, and pass that variant to your children. Over many generations, that variant becomes more common in the population. For physical traits like skin color, the connection is direct. Lighter skin in low UV environments means better vitamin D synthesis, which means stronger bones, better immune function, and higher survival rates. The evolutionary pressure is clear and consistent, but cognitive ability doesn't work that way. Intelligence, however you define it, is useful in every environment. A hunter in the Amazon rainforest needs spatial reasoning, memory, and problem-solving skills just as much as a farmer in ancient Mesopotamia or a fisherman in Polynesia. There's no environment where being smarter is a disadvantage, and there's no environment where a specific type of intelligence is uniquely beneficial. Think about the challenges faced by early human populations in different parts of the world. Arctic populations had to track migrating animals across featureless ice fields, predict weather patterns, and engineer complex clothing from animal skins to survive temperatures that would kill an unprotected human in hours. Desert populations had to memorize the locations of scattered water sources across hundreds of miles, navigate by stars, and understand the behavior of dangerous animals. Rainforest populations had to identify hundreds of edible versus poisonous plants, track prey through dense vegetation using subtle signs, and maintain complex kinship networks across dispersed communities. These challenges are different in their specifics, but they all require the same underlying cognitive toolkit: working memory to hold and manipulate information, executive function to plan and adjust strategies, social intelligence to cooperate and share knowledge, pattern recognition to learn from experience, language to transmit that learning to the next generation. Every environment demanded all of these abilities simultaneously. There was no cognitive trade-off where you could sacrifice one skill to excel at another. In fact, the evolutionary pressures that shaped human cognition were largely social, not environmental. The ability to cooperate in groups, communicate through language, understand social hierarchies, predict the behavior of others. These pressures were universal. Every human society, regardless of geography, faced them. So the genes that support these abilities evolved in all populations simultaneously. Anthropologists call this the social brain hypothesis. The idea that the primary driver of human intelligence wasn't tool use or environmental adaptation, but the need to navigate increasingly complex social relationships. As human group sizes grew from bands of 20 to tribes of 150 to chiefdoms of thousands, the cognitive demands of tracking, who owed favors to whom, who could be trusted, who was rising or falling in status, those demands exploded. The individuals who could handle this social complexity were the ones who survived and reproduced. And this pressure was identical across all human populations. This is why the genetic variation within populations is so much greater than the variation between populations. Your cognitive potential isn't determined by your ancestry. It's determined by the random combination of alleles you inherited from your parents. Two siblings, same parents, same ancestry, can have dramatically different cognitive profiles. One might excel at verbal reasoning while the other excels at spatial tasks. The genetic differences between them can be just as large as the differences between individuals from opposite sides of the planet. The Broad Institute study also debunked another persistent myth: the idea that certain populations have evolved superior abilities in specific cognitive domains. You've heard the stereotypes. Asians are better at math. Europeans are better at abstract reasoning. Africans are better at physical tasks requiring quick reflexes. These ideas have been repeated so often that many people accept them as fact. The genetic data shows no evidence for any of this. The genes associated with mathematical ability, spatial reasoning, verbal fluency, they're distributed equally across all populations. Yes, there are cultural and educational factors that influence performance in these areas. A child who grows up in a society that emphasizes mathematical training will likely perform better on math tests than a child who doesn't. But that's environment, not genetics. The researchers went a step further. They looked at ancient DNA from Neanderthals and Denisovans, extinct human relatives who interbred with Homo sapiens tens of thousands of years ago. Modern humans of European and Asian descent carry small amounts of Neanderthal DNA. Some populations in Oceania carry Denisovan DNA. Did this archaic DNA contribute to cognitive differences? The answer was no. The Neanderthal and Denisovan genetic variants that survived in modern humans are mostly related to immune function and skin traits. Very few are located in genes associated with brain development, and the ones that are show no pattern of enhancing or diminishing cognitive ability. If anything, the archaic DNA we carry is functionally neutral when it comes to cognition. This makes sense from an evolutionary perspective. Neanderthals and Denisovans weren't less intelligent than Homo sapiens. Archaeological evidence shows they made tools, controlled fire, created art, and buried their dead with ritualistic care. They were cognitively complex. Interbreeding with them didn't create a cognitive advantage or disadvantage. It just added genetic diversity. So if the science is this clear, why do the myths persist? Why do people still believe in genetic hierarchies of intelligence? The answer is partly historical. For centuries, those in power used pseudo-scientific theories to justify slavery, colonization, and genocide. If you could convince people that certain races were biologically inferior, you could rationalize treating them as less than human. The science was always garbage, but it was politically convenient garbage. Even after the civil rights movements of the 20th century dismantled legal segregation, the underlying assumptions lingered. They showed up in education policy, in hiring practices, in criminal justice, in media representation. The idea that intelligence is tied to race became so embedded in culture that it felt like common sense, even without evidence. The other reason is that humans are pattern-seeking creatures. We see differences in outcomes, educational attainment, income levels, test scores, and we want an explanation. Genetics feels like a simple answer. It absolves us of having to confront uncomfortable truths about systemic inequality, access to resources, and historical injustice. If the gaps are genetic, then they're inevitable. Nothing to be done. But the science doesn't support that narrative. The gaps are real, but they're not genetic. They're the result of centuries of unequal access to education, healthcare, nutrition, and opportunity. When you control for those factors, the gaps shrink or disappear entirely. The Broad Institute study included a secondary analysis that illustrated this perfectly. They compared the brain gene expression patterns of individuals from different socioeconomic backgrounds within the same racial group. What they found was that poverty, chronic stress, and lack of educational stimulation during childhood actually altered gene expression in the brain. Not the DNA sequence itself, but which genes were turned on or off, a field called epigenetics. The mechanism works like this. Environmental factors can attach chemical tags to DNA or to the histone proteins that DNA wraps around. These tags don't change the genetic code, but they do change how accessible that code is. A gene that's tightly wrapped and tagged might never get read by the cellular machinery that builds proteins. A gene that's loosely wrapped and untagged might be read constantly. Same DNA, radically different outcomes. Children raised in environments with high stress and low resources showed different patterns of gene expression in regions of the brain responsible for memory and emotional regulation. Specifically, they showed altered expression in genes related to cortisol receptors, the proteins that help the brain respond to stress hormones. When you're exposed to chronic stress as a child, your cortisol system goes into overdrive. The epigenetic tags respond by dampening the expression of cortisol receptors, essentially desensitizing the brain to stress signals. This adaptation helps in the short term. It prevents the brain from being overwhelmed. But in the long term, it can impair memory formation and emotional regulation. The same children also showed differences in genes related to neuroplasticity, the brain's ability to form new connections and adapt to learning. Genes like BDNF, brain-derived neurotrophic factor, which acts like fertilizer for neurons, showed reduced expression. Less BDNF means slower learning and weaker memory consolidation. Again, this wasn't genetic destiny. It was environmental impact on genetic activity, and it was reversible. When those same individuals gained access to better resources and opportunities later in life, some of the gene expression patterns normalized. The epigenetic tags could be removed or modified. The brain's plasticity, the very thing that had been suppressed, allowed it to recover when the environment improved. This is why early intervention programs, things like Head Start and high-quality preschool, have such dramatic effects. They're not fixing genetic deficits. They're removing environmental barriers that alter gene expression. This is the real story the data tells. Your brain's potential isn't locked in by your ancestry. It's shaped by your environment, your experiences, and the opportunities you're given. Genetics provides the raw material, but environment and culture build the structure. The implications of this research go far beyond academic debates. They challenge the foundation of scientific racism. They dismantle the justification for discriminatory policies. They force us to confront the fact that the disparities we see in the world aren't inevitable. They're the product of choices, systems, and histories that can be changed. Some critics have pushed back against the Broad Institute findings. They argue that the study didn't look at every possible genetic variant, that there might be rare variants in specific populations that weren't captured. Technically true, but the burden of proof is on those making the claim of difference, not on those demonstrating similarity. And after decades of research and hundreds of studies, no one has found credible genetic evidence for cognitive differences between races. Others argue that even if the average genetic potential is the same across populations, there might still be differences in the distribution. Maybe one group has more individuals at the extreme high end of cognitive ability. But again, the data doesn't support this. The distribution curves overlap almost completely. For every genius in one population, there's a statistical equivalent in every other population. The most important takeaway from this research isn't just what it tells us about genetics, it's what it tells us about ourselves. We are one species. We share 99.9% of our DNA. The remaining 0.1% accounts for all the visible diversity we see: skin color, hair type, facial features. But when it comes to the invisible traits, the ones that shape how we think, learn, and solve problems, we're fundamentally the same. That 0.1% of genetic difference is worth understanding in more detail. It represents about 3 million base pairs out of the 3 billion that make up the human genome. Most of those differences are in what's called junk DNA, regions that don't code for proteins and have no known function. Of the differences that do occur in functional genes, the vast majority affect traits that were under strong environmental selection pressure in the past 70,000 years: skin pigmentation, lactose tolerance, altitude adaptation, disease resistance. The differences are real and important for understanding human health and history, but they don't extend to the architecture of cognition. This doesn't mean we're all identical. Individual differences in cognitive ability are real and measurable. Some people have better working memory. Some have faster processing speed. Some excel at creative thinking. These differences matter. They shape our lives and our choices. But they're not predicted by race. They're not the result of ancient evolutionary adaptations. They're the product of the unique genetic lottery each of us inherits and the environments we grow up in. To illustrate how much individual variation outweighs population variation, consider this. If you take two random people from the same ethnic group and compare their genomes, you'll find hundreds of thousands of genetic differences between them. Now, take two people from different continents, as genetically distant as possible, and compare them. You'll find maybe an additional 10 to 20,000 differences beyond what you found within the first group. The between-group difference is a tiny fraction of the within-group difference, and almost none of those extra differences touch the genes involved in cognition. The Broad Institute study is still being cited in ongoing research. It's being used to design better educational interventions, to challenge discriminatory practices in hiring and admissions, and to inform public health strategies that address the real causes of cognitive disparities. What these researchers discovered in the genetic code wasn't a hierarchy. It was a mirror, a reflection of the single human family that migrated out of Africa and adapted to every environment on Earth without losing the cognitive abilities that truly define us all. The differences we obsess over, the ones we use to divide ourselves, they're skin deep. And maybe that's the most shocking result of all. Not that we're different, but that we are all the same.